With a diameter of 1.2 to 2.4 millimeter and with 4 to 6 mm microemulsifier. So this is one of the definitions. You may not agree with this, but like scientists, they are trying to, just like for the mesenchymal stem cells, there are a lot of different definitions. For SVF, there are a lot of different definitions. Nanofat there are also, but I must clear here that nanofat is not cell-free. It do contain the cell, that's why it's very effective. So there are like tons of studies on these kind of like procedures. These could be enzymatic procedures for different applications, for breast augmentation, for erectile function, for beautification, for the face rejuvenation, and so many procedures for osteoarthritis. And so these studies are available, so you can optimize the protocol. Like there is a clinician who is going to do for osteoarthritis. There are tons of studies available, so we can optimize the protocol for you. One thing that Dr. Fidon, he has mentioned that you can have a setup. I must say that with a clinician, you should have a basic scientist with you, so he can also optimize the protocols for you. He can do the better preparation because his life is do the pipetting, do the microscopy and so on. Clinician, they can do their own role, like what patient we have to treat, how we have to treat, and so on. So this could be a good combination, and I must appreciate Dr. Bushra's effort that she has like combined the researchers and basic scientists. They are working together for the benefit of our patient. So like the things are going forward and beyond, because nowadays not stromal vascular fraction SVF or nanofat, but total stromal cells. This is the new definition, TOS. So now we are talking about the TOS. So it's a combination of SVF and fat. So you can use both for like osteoarthritis and many other applications. And there are so many studies available now. So this is the future, and things are being modified. We should, as Dr. Ali has mentioned, that we should be very close to the literature. We should read the studies. So this is how we can read different studies, and we can go for what are the advancements they are taking. So, for example, there was discussion for the face, like if we have to do the face rejuvenation or face uplifting. There are different, like for the chin, for example, we can have a different fat combination, like for diagnoser, as you explained. We can have a 1200 micrometer fat, and we can use there. For like eyelids and for forehead, we can have a different combination and different like volume of that. So it's actually, as he mentioned in the PRP, just like our aesthetics, they should have a better understanding that what fat they are going to use. Either it has PPP, it has PRP, how much volume we are going to use. But we can't use the same prep in the whole face. It can have the complications. So we can have, like this is now more and more rotations being available, so we have to modify the procedure. So there was also discussion by Dr. Falsi explaining that how our procedure SVF can help the hair regeneration, so I'm not going to talk about this. So, but this is like very helpful. There are so many studies available that how we can regenerate the hair using these kind of technologies. The next thing is coming. Now we have recent collaboration with National University of Singapore, and we are going to work on next generation stem cell. This is stem cell 2. Stem cell 2 is we do the modification in the stem cell. For example, in this case, we are going to use the glioblastoma with the neuronal stem cell. So in case of neuronal stem cell, what procedure we used to do initially, we used to modify the cells with the microbiologists, and Professor Fidoni is very much familiar with these kind of technologies. What we are going to do next, like there are a lot of restriction if you use the viruses for the modification purposes. What we are going to do next, we are using the vectors with these kind of, so these, the DNA is not going to integrate into the stem cell. There was problem of integration, so we are not going to do that. And these are, we have like our collaborator in Singapore, they have got very good results, and we are also trying to have some clinical trials in Pakistan. So this is the future. As he had mentioned, in future, our clinicians will be the cell biologists. They are going to treat the patient with the cells and cell products. So similarly, the future is the regenerative medicine. In each application, the diseases that we don't have any cure. For example, Parkinson's is there, Alzheimer's is there, and so many other diseases are there. This is a hope for the patients, and if we will work together, the clinicians and the scientists, we can bring not only hope for the patients, we can actually treat those patients and they can have a better life and better future. Thank you very much for your listening.